In experiment.
We don't know yet.
Why is light so fast?
Like, it sounds so simple.
They had no idea.
But now the data speaks.
I find this not only refreshing, but at some level astounding.
Nature.
Welcome back to the Nature Podcast. This week, the AI that identifies gene interactions
and assessing the impact of rocket noise on wildlife. I'm Nick Petrichow.
and I'm Benjamin Thompson. This week, Nature has a paper by a team of scientists who have developed a way to use an artificial intelligence system to predict the interaction between genes in human cells. They hope that it will speed up the mapping of gene networks and have already used it to identify genes that are potential therapeutic targets for a type of heart disease. Reporter Alex Lathbridge takes up the story.
Going on inside your cells right now are a countless number of biological processes that are essential to things like the development and maintenance of healthy tissues. These processes are regulated by a complex system of genes. In many cases, right now at least, researchers don't know how they're all connected. And figuring out these networks is no easy task, as Christina Theodoris from the Gladstone Institute of Cardiovascular Disease in the US explains.
Let's say there was only two genes in any cell and you were only looking at those two changes, then you could pretty easily see what happens when one goes up and the other goes down. But when you start looking at 20,000 genes that are within the genome, that's when it becomes much more complicated, because many of these genes are wired in a way that they affect downstream genes by increasing or decreasing their expression. Those connections can change in different states across time and development or space within the body and different tissues. They can also be rewired in disease states. And all of these many genes interacting in a coordinated way becomes a very complex process that's difficult to untangle, which is why we've turned to artificial intelligence and machine learning to help us.
This week, Christina and her colleagues have published details of an artificial intelligence system called GeneFormer that they hope will speed up the process of identifying how genes interact. But to allow GeneFormer to learn the fundamentals of how genes interact within networks, it had to be trained on a huge dataset. In this case, publicly available data on the transcriptomes taken from 30 million cells.
Transcriptomes are data sets where we've measured the expression level of each gene in the genome, which tells us how active or inactive the gene is. And these data sets are measured in individual cells from different cell types, developmental stages, or disease states. And so by collating all of these individual cells into a large-scale data set, we can then train the model to understand how these gene networks change to control the state of the cells in each of the different settings. The model, through a completely self-supervised way, was able to learn to pay attention to genes that were really central within gene regulatory networks, and without having previously been told that these are transition factors or central regulators within a network. It was able to learn that on its own.
And the team could use GeneFormer to virtually ask questions of simulated gene networks.
We can take cells, take the set of genes that are expressed there, and delete one of them, and then ask, what does that do to the gene network within the cell, and what does that do to the cell state? So, for example, if we take a healthy cell and we delete a gene, does it move to a particular disease state? Or vice versa, which we refer to as in silico treatment analysis. Can we take a disease cell, delete a gene, and see whether that moves it back towards the healthy state, which would indicate that that gene could be a candidate target for a therapeutic intervention.
Central to GeneFormer's function is a process called transfer learning, where the AI can apply information it's learned about one situation to another one. Think of it like learning to drive a car. You train enough and you can drive it. Then, without too much difficulty, you can transfer that learning to drive a van. So one of the team's key objectives was to see how GeneFormer's learning based on a huge dataset showing the gene activity inside cells, could be applied to situations where activity data is sparse, and where this in silico treatment analysis, predicting how removing a gene could affect a diseased cell, could have real impact. And so their focus turned to cardiomyopathy, a group of diseases that affect the heart muscle.
And the reason we were interested in studying cardiomyopathy specifically is because it is a progressive disorder. So we would have the opportunity to intervene and prevent the progression if we had a targeted medical therapy. And this can make huge impact in the lives of these patients that often progress to heart transplant when their heart fails. And because it's a relatively rare disorder and affects the heart tissue, which is hard to sample in the clinic, there are limited data sets specific to this disease. So when we applied the approach of transfer learning, we were able to predict candidate genes whose targeting would improve the disease, even though we only had limited data specific to cardiomyopathy. And in doing that in silico, we were able to do so much more efficiently, both with regard to time and cost, to really come down to a prioritized list of predictions that we could then test in the lab more easily.
Geneformer came up with a list of priority genes, ones which it predicted would improve the state of the cell if removed from the network. In the lab, the team set about testing heart muscle cells, known as cardiomyocytes, to see if inactivating the genes could make a difference. And they found something.
We were able to identify candidate therapeutic targets that, when we tested them experimentally in the lab, did indeed improve the function of contracting of these heart muscle cells. Additionally, we were able to identify a gene that was not previously known to be key for cardiomyocytes, where when we knocked it down, it disrupted their ability to contract. So we were able to use this transfer learning approach to make new discoveries and biological insights.
While there is some transcriptome data specific to cardiomyopathy, this has come from cells taken in heart biopsies, generally when patients are at a later stage of the disease. So one of the goals of the lab is to create a model that can help identify the potentially important therapeutic targets at earlier stages of the disease, allowing intervention to make a bigger impact. And while Geneformer's predictive power is promising, Christina and the team know that when it comes to mapping complex networks, things are complicated.
When you are testing these changes in the model, you are really only modeling that first step of the change. So removing that gene, does it shift the cell towards the state of interest? So you're looking at the direction of the shift. In reality, if you were to remove that gene, there'd be many downstream consequences that would occur over time that would change the cell more and more until it became to that downstream final state.
To address this, the team are working to develop a model that could help build a better picture of how the network progresses after those first changes are made. But despite some of its current limitations, Christina and the team think that the approach that they've taken with Geneformer could be a powerful tool for researchers more broadly.
So I think one thing we want to emphasize is that by seeing a very broad range of human tissues, the model is able to improve predictions in many different applications and different settings, not just cardiovascular disease that we particularly were interested in. And there's a lot of potential in researchers using this to answer questions in their fields and diseases of interest.
Christina Theodoris there. For more on this story, head over to the show notes for a link to the paper.
Coming up, how researchers are going to figure out the effect of rocket noise on wildlife. Right now, though, it's time for the research highlights with Dan Fox.
Researchers have found and identified bacteria that could help break down some of the forever chemicals accumulating around the world. PFASs, per- and polyfluoroalkyl substances, are a group of widely used synthetic compounds that have been nicknamed forever chemicals because of their incredible persistence in the environment. This longevity comes because PFAS molecules are full of fluorine atoms, which bind tightly with carbon, forming bonds that can't be easily broken down by microbes in the environment. But chlorine carbon bonds are weaker, So the team behind this new research wondered if the chlorine atoms in some forever chemicals might give microbes a point of attack. The researchers obtained microbe-rich sludge from a local sewage treatment plant and exposed it to a variety of chlorine-bearing PFAS. They found that after the bacteria first removed the chlorine, they could then remove almost 80% of the fluorine atoms. Finally, they used genomic sequencing to identify the species of microbes most closely related to those in the sludge. The team hope that their findings will lead to new chemical cleanup technologies and perhaps the creation of biodegradable versions of forever chemicals. You can read that research in full in Nature Water. When it comes to gummy sweets, texture is just as important as flavour. Now, scientists have used some clever chemistry to identify the precise combination of ingredients needed to ensure that gummies keep their chew. If gummy sweets are stored for too long or in the wrong conditions, they can become harder and less satisfying to chew. In an effort to maximise how long they stay fresh and soft, researchers tweaked the factors that most affect texture, the recipe's concentrations of starch and gelatin, and the ratio of glucose syrup to sucrose. The team measured the properties of the resulting sweets during storage for different lengths of time and at a range of temperatures. They then used the sweet's mechanical properties to estimate how aging affects a crucial feature: the average distance between its molecules. Sweets with longer intermolecular distances are softer. They also estimated freshness by measuring other physical characteristics, such as water content and pH. Out of the 8 recipes studied, the team found that 3 resulted in optimal shelf life, including one of them which surprisingly involved no starch at all. You can chew over that research in Physics of Fluids.
Finally on the show, it's time for the briefing chat, where we discuss a couple of articles that have been highlighted in the Nature briefing. Ben, what have you got for us to chat about this week?
Well, I've got a story that I read about in Nature, and it's about some research that's helping to explain why chronic stress can inflame the gut, and it could have important consequences in how this inflammation is treated.
Well, I think I've heard things like this before, so maybe give me a little bit of a refresher. How does stress affect the gut?
Yeah, it's true that psychological stress is known to worsen gut inflammation, particularly that caused by certain bowel diseases, like inflammatory bowel disease. And quite why this is hasn't really been well understood. And these conditions can be very serious, right? Inflammatory bowel disease has a bunch of symptoms: abdominal pain, diarrhea, and fatigue, to name just 3. And the 2 main types of IBD are ulcerative colitis and Crohn's disease. Okay, and these can be debilitating for people who have them. And it's known that stressful events often precede IBD flare-ups. Okay, there's an association between the two. But really why that is hasn't been particularly well understood, and it seems like researchers have found a link between the brain and the guts.
So there's a link there, but we're not quite sure how it works. But does this new study tell us a little bit more about what exactly is going on here?
Yeah, they have an idea, and they've studied it in mice, and these mice have IBD-like symptoms. And this pathway, as I say, starts in the brain and ends in immune cells in the gut in particular. And so what they found is that after a surge of stress, the brain sends signals to the adrenal glands, and these release chemicals called glucocorticoids. And these are absolutely key to this whole story, okay? And these are released to the rest of the body.
Right, okay. So how are the researchers trying to figure out what these glucocorticoids are doing?
Well, initially the article said that the researchers thought that these glucocorticoids were directly affecting immune cells in the gut, right? But actually they showed there's a little bit more to it. So actually what's happening is these molecules are making their way into the gut, and they're acting on neurons in the gut, and in particular a specialized type of neuronal cell called a glial cell. And when activated, these themselves activate the immune cells. Okay, so we're getting to this sort of end game now. And the immune cells release these special chemicals that are usually designed to ward off pathogens. But in this case, there are no pathogens present. So what it does is it leads to the inflammation in the gut, which is a sort of key symptom of IBD, and that can cause a great deal of discomfort for people.
Now understanding this pathway a bit better, does that leave the door open for sort of new therapeutics and things like that?
Well, actually, it leads to a bit of a paradox, an unexpected paradox. So one of the treatments that's used for IBD is glucocorticoids.
Oh.
Okay. Yeah, I know. So, and this is something that the researchers are trying to figure out, to be honest with you. And they say in the article, this came across as a big surprise. And what maybe seems to be the case, but sort of TBC, I guess, is that it's about timing. So when someone's treated with glucocorticoids, that's quite a short-lived treatment. And in a short time, it seems like these molecules might have an anti-inflammatory effect. But when it comes to things like stress and this kind of chronic levels of stress that are sometimes associated with IBD, this longer time frame seems to flip things on its head and turn this into a pro-inflammatory situation. But as I say, this is currently in mice and there's lots to understand.
So there is this sort of paradox then. Where does that leave the research? What are the sort of next steps now?
Yeah, well, it shows something that I think is important, that there's more evidence of the brain's ability to drive inflammation in kind of distant parts of the body. And I think we've covered on the show how, brain and body are so interconnected when it comes to disease states. And they say that perhaps in future this research means that treatment will need to combine drugs maybe with stress management techniques, and that could be more effective than just drugs. And potentially knowing more about this pathway could mean new therapeutic targets that go for just it. And there's also evidence that stress can cause inflammation in potentially other parts of the body as well. So maybe by knowing a bit more about it in this situation, it could offer new insights into how the brain and body are connected in situations like that.
Well, obviously there's lots more to understand here, but hopefully in the future we'll hear more about it and possibly new therapeutics for people who have these conditions. For my story this week, I've been reading in Nature about how noisy rockets are and how researchers are trying to understand how the noise from their launches is affecting wildlife.
Well, I spoke to a researcher a couple of weeks ago who was at the launch of the JWST in French Guiana, and they said that there was this impossibly loud rumble for a bit as the rocket took off. Is this the sort of place they were looking at?
Yeah, I mean, this article focuses on a couple of launch sites in the US. So there's one in Texas, one in Florida, and one in California, and they're all in biodiversity hotspots within the US. So they're far away from people and obviously close to wildlife. So there could be a big impact. And as you said as well, they're very noisy. So, for example, the launch of Artemis 1, which you probably remember, was recorded to be 127 to 136 decibels, which is louder than a clap of thunder, from several kilometres away. So it's pretty loud, and so it's understandable to think that there may be effect on wildlife, but the reality is we don't necessarily know what that is.
So what sort of things are researchers wanting to know?
Well, they want to know if such noise is having an effect on animals' behaviour, whether it's causing them stress, or whether it's affecting things like birdsong. For example, if you and I hear a loud noise, we may shout to compensate. Do birds do a similar thing with their song, and would that have an effect on them? So they want to understand what the effects are, and this is especially pertinent now because the number of rocket launches is increasing substantially. So, for example, in this place in California where a lot of rockets are launched, called Vandenberg, it historically had about 5 to 15 rockets launching per year. But by 2030, that's going to go up to 50 to 100 per year. So there's a substantial increase in the number of rockets that are going up, and that may well be having an effect on the wildlife, and we need to understand that.
And how best to sort of go about looking at what that effect might be then?
Well, the researchers are doing a 3-year study where they're using cameras and microphones to assess the impacts on the behaviour. So things like the bird song and how the animals are reacting. Do they look, startled, or what's their sort of reaction to these very loud noises? And to have some sort of long-term monitoring to see if that has any impact on the species around there, especially some of the endangered or protected species that exist around these sort of launch sites.
And do they suggest any things that could be done to maybe mitigate these effects? Obviously, we don't know what they're going to find yet, but what have they put forward?
So they've got funding for 3 years, but they're actually going to try and do this for more than a decade. But As it stands, rockets already have an amount of sound dampening that goes on. So if you've ever watched a rocket launch, you may be familiar with the big fire trenches that are underneath when a rocket goes off, and this sort of channels where the rocket blast goes. And this also helps to sort of dampen the noise. And also for many launches, they use a lot of water underneath the rocket to absorb a lot of noise. So things like this could be done or changed. Or the other thing is they may identify sensitive periods when the wildlife are particularly susceptible to a lot of noise. Maybe it's their breeding season or something like that. And that could inform whether rocket launches should be scheduled at a different time, like to avoid this sort of very sensitive period for them. So it'll be interesting to see how this research shapes up going forward and what that may mean for how we launch our increasing number of rockets.
I mean, I suppose we do talk about rocket launches a lot on this podcast, but really we're thinking about what happens after that has happened. And I think really thinking about what's going on at the time as well is an important avenue that I maybe hadn't fully considered. So yeah, very interesting to see where this one goes. But let's leave it there for the briefing chat this week. And listeners, for more on these stories and where you can sign up to the Nature Briefing to get even more like them delivered directly to your inbox, check out the show notes for some links.
And that's it for this week. Just before we go, don't forget you can keep in touch with us on Twitter. We're at Nature Podcast. Or you can send us an email to podcast@nature.com. I'm Nick Percher-Chow.
And I'm Benjamin Thompson. Thanks for listening.